Thousands of years ago in sub-Saharan Africa, people were battling a dangerous threat, the tsetse fly. This wasn't just any fly. When it bit you, it could inject a tiny parasite that caused sleeping sickness, an illness that messed with your sleep and eventually killed you. Over time, some people's bodies developed a clever genetic change, a clever and random genetic change, a special version of a gene called APOL1 that acted like a shield against this disease. This natural defense has been saving lives ever since, especially because the fly and the sickness it transmits is still around today. But here's the negative part. Here's the twist. Our body's genetic solutions sometimes come with unexpected side effects. These APOL1 genetic variants that protected against the deadly sleeping sickness also increase the risk of developing preeclampsia, a serious pregnancy complication that causes dangerously high blood pressure. Preeclampsia can lead to babies being born too early and is the second highest cause of pregnancy-related deaths worldwide. While scientists have known for about a decade that APOL1 gene variants can increase risk for kidney disease, their link to preeclampsia risk is new, having only been illuminated in the last 5 or so years, with more questions than answers surrounding their role. One of the key scientists asking and attempting to answer those questions is Dr. Wendy Kuohung. This February, March of Dimes awarded Dr. Kuohung a 2025 Discovery Research Grant, providing her $200,000 to hunt for treatments for preeclampsia connected to the two APOL1 gene variants, called G1 and G2. As part of her research, Dr. Kuohung is building something pretty cool, a test where she can take placental cells that have these APOL1 variants, expose them to different medications, and see which ones help reduce stress inside the cells. But she's not stopping there. She'll also be the first scientist to watch exactly how these variants trigger preeclampsia in placental cells, kind of like a detective observing a crime as it happens instead of just investigating afterward. Her research is a bit of a twofer. First, she could discover new medicines that might help treat preeclampsia. Second, she'll help all scientists better understand how preeclampsia works in Black women who carry these variants. What makes Dr. Kuohung particularly suited for this work is that she's both a doctor who treats patients and a scientist who works in the lab. She's doing bench-to-bedside research. She's at the forefront of something big, and she's here today to tell us all about it. This episode is about APOL1-related preeclampsia and the discovery of drugs to treat it. Stay with us. Dr. Wendy Kuohung is a 2025 March of Dimes Discovery Research Grant winner, an associate professor of obstetrics and gynecology at Boston University Chobanian and Avedisian School of Medicine, and the director of the Division of Reproductive Endocrinology at Boston Medical Center. Dr. Kuohung joins me from Boston. Dr. Kuohung, are you in Boston? Or are you outside of Boston technically? No, you're in Boston.
Yes, I'm in Boston.
I'm technically outside of Boston, but I'm in the suburbs. So welcome to the show.
Oh, it's great to be here, Alessia. Thanks for having me.
Of course, it's good to have you here. Now, before we talk about the work you'll be doing as part of the Discovery Grant, I want to ask a question about your other job. You are a fertility doctor. We mentioned that you're head of REI at Boston Medical Center. You're a fertility doctor. You help people get pregnant every day. Now, but you don't technically deal with preeclampsia in those first 9 weeks of pregnancy, right? You deal with getting pregnant and implantation and HCG and making sure that the tubes are not blocked and all this kind of stuff. So why is it that you're passionate about preeclampsia when you don't actually deal with this in your 9-to-5 job as a physician?
Yes. So great question, Alessia. So one of the treatments that I use a lot, one of our flagship treatments, is in vitro fertilization. And we know from many studies that doing in vitro fertilization on patients increases their risk of preeclampsia and the resulting pregnancy. It's not really well understood why the increase for the patient's risk for preeclampsia is increased, even outside of their age-related risk. And then there's some thoughts that it may have something to do with, the presence or absence of a corpus luteum in things like frozen embryo transfer cycles. But we do know that just doing in vitro fertilization increases a patient's risk for preeclampsia. So, this research, if it increases our understanding of the causes of preeclampsia, that might also help us find out how to decrease the risk associated the risk of preeclampsia associated with in vitro fertilization.
Yeah. How just quickly, how new is the understanding that IVF increases preeclampsia risk, and how high is that increased risk? Is it significant?
Yeah, I mean it's a small percentage increase, but it is been shown to be significant, and I would have to say it's probably been recognized probably over the last decade that
Okay.
there's been an increased risk.
Okay. All right. So now I understand why you're interested in, and you're also interested in the placenta too, right? You've got a separate kind of placental interest.
Yes, yes. I've always been interested in implantation and how the placenta develops, and what are the factors that interfere with placental invasion.
Yeah, and we know that preeclampsia has a lot to do with the placenta. So there
Mhm.
we've come full circle. Now I have some questions about this variant, Wendy. When you told me about this fly, because you're the one that clued me into this whole thing, the sleeping sickness and the APOL1 variant and all that, you told me about this fly that carries the parasite that
Mhm.
the APOL1 gene variant fights. I was really fascinated. I looked it up, and yeah, I saw that this fly is still doing this. It's still causing sleeping sickness, and it became known as sleeping sickness because the fly interrupts sleep and other terrible effects too. And like I mentioned earlier, it can be fatal. So my first question is about this variant. Why did only some people develop the variant, the APOL1 variant? Why didn't everyone develop this protection against sleeping sickness in that region?
Right. Well, the issue is that sleeping sickness, like malaria, is not universally fatal. So the selection for the APL1 variant was it favored people who carried the APL1 variants, that helped them, gave them a survival and reproduction advantage. But because not everybody, it's not universally fatal. There's still, you know, individuals who don't have the variant who are going to survive, and not necessarily succumb to that disease.
So does that mean that people who were born as a result, people who had family members that died from sleeping sickness ended up developing the variant, while others who kind of weathered the sickness better and didn't die, they didn't develop it? Is that how it worked?
Well, it's thought that there are two main variants for APL1 that arose probably
Yes.
around 10,000 years ago, and
Yeah.
so there was a survival advantage for people who did carry the variant, but it wasn't a universal thing that it didn't necessarily mean that if you didn't have the variant that weren't going to survive, but there was probably a reproductive advantage for those who did carry the variant because they were more likely to survive when there were outbreaks of the illness.
Okay. Okay. Understood. And so basically, you didn't need the variant to survive, and so that's why not everyone developed it, because it
Right,
wasn't
right.
actually essential for survival.
Yes,
Okay.
yes.
And now for the people that don't have it, they
Hmm.
just have the gene, right? But just not the
Yes,
variant. So
right.
you and I, we have the APOL1 gene.
Yes, yes.
Everyone on the planet.
Exactly. And the variants were are mainly seen in patients with of recent African ancestry. So in certain parts of sub-Saharan Africa. So it's going to be mainly seen in the U.S. population. Is mainly going to be seen in Black individuals of recent African ancestry.
Okay, so we're not talking here about all Black people in the world. We're not
Right.
talking about all African people. We're not talking about all African American people. We're talking about um, either people currently in sub-Saharan Africa, some of them, not all of them, or pretty recent, within the last 100 years or so, emigres from
Yes.
sub-Saharan Africa to various parts of the world,
Yes.
including America.
Yes. And in fact, there was a recent study that looked at like place of birth and in Boston, the Boston birth cohort. They saw that patients from Haiti and then patients who and African Americans, that the patients from Haiti actually they're looking at the APL1 genotypes in the placentas and the patients, that they did not show increased risk for that carrying the APL1 variant, whereas in patients, so the ones from Haiti did not really have a high proportion of APL1 variants, whereas the African American patients did show that there was a higher incidence of the APL1, or higher prevalence rather, because there was transport of individuals from Africa to the Americas, probably to Haiti and the U.S. were probably around the same time. So
Okay.
it was probably more of the location in Africa where they came from that may have not had a
Oh,
high prevalence of that variant.
so we know that there are some Black people around the world that have this. We know some Americans have this as a result of being immigrants from sub-Saharan Africa. And so you told me that approximately 50% of African Americans carry at least one variant of this gene. So they carry either G1 or G2, and 13% carry two.
Yes.
Now, talk to me about the genotype here, because you told me that this type of preeclampsia, APOL1 related preeclampsia, is determined by the fetal genotype, not the maternal genotype, which means the baby that the woman is carrying dictates the risk for the mother, not the mother's genotype. So, so tell me about how that would work with G1 and G2 and what the baby is carrying.
Yeah. So they epidemiologic studies that looked at genotypes of the placenta, which is the fetal genotype, and compared to the maternal genotype, found that the risk of developing preeclampsia was more dependent on the fetal genotype if the fetus carrying one or more of the APL1 variants, and they also found that not every single pregnancy where the fetus carried an APL1 variant developed preeclampsia. So there was probably also some second hit needed, like either some environmental insults or triggering factor, like maybe an infection that would be needed for the pregnancy to actually, develop preeclampsia.
Yeah. Do we know what the risk of preeclampsia is with one variant versus two variants?
Yeah, so it's increased to. I mean, we don't know. There's more studies going on right now. We don't know exactly how much it's increased, but it's probably at least a doubling of the increased risk of preeclampsia if the fetus carries an APL1 variant.
Mm-hmm. If they only carry one.
Yes.
Okay, so there's a doubling of the risk from 0 to 1. We
Mm-hmm.
don't know exactly how much higher the risk goes from 1 to 2, but we know to definitely get preeclampsia, the fetus has to have. Now,
Yeah.
Wendy,
Yes.
could there be one variant in the fetus and a
Mm-hmm.
second hit that would lead to preeclampsia, or you need two variants plus a second hit?
No, they can carry a single variant, and then but
Okay.
it's more of a risk if there's two, and we know that the G2 variant is higher risk than the G1 variant.
Okay, so they could technically carry just G2 and then they would need a second hit. Now, this is really interesting. So, genetics alone is not enough to give you preeclampsia here based on what we understand. Okay, but you can still get preeclampsia with only one variant and
Yes.
a second hit, or two variants and a second hit, which means another factor. Tell me about what these factors could be. You mentioned infection.
Mm-hmm.
Is that like a bacterial vaginosis infection or like a group B strep infection? And what other type of insults could cause this final blow to the
Yeah.
risk ratio?
Right. Well, it's thought that some infections might cause increase the risk of preeclampsia in patients whose fetus carries APL1 variants, possibly due to issues with increased interferon, or possibly a decreased ubiquitization of the APL1 gene. So there's different possibilities for the second hit. They haven't identified definitively what the second hit is, but because not all patients whose fetus has a high risk APOL1 genotype develops preeclampsia, we know that there's some additional factor that's required for the development of preeclampsia, and not just the presence of the gene variant.
Yeah. And so thinking further ahead with the work that you're doing to discover these drugs that could treat preeclampsia, are you going to give, theoretically, hypothetically, years down the line when this is clinically available, hopefully it will be, and obviously we wish you all the success in your work. Is this something that you will give to women proactively that have one variant only? Because we don't know what that final hit is. We don't know if she'll actually develop preeclampsia. So is this something that you're going to give to anybody that has one variant in the fetus? And how early can that be detected? How early can we detect fetal genotype? Probably immediately at pregnancy.
So there haven't been any early studies looking at APOL1 genotype of fetuses, although in my reading I did come across that some groups in Cleveland were proposing that APOL1 gene testing be done, possibly in as part of first trimester screening and cell-free fetal DNA testing.
Okay. So around 10 weeks.
So the hope is that if we can find a way to interfere with the development of APOL1 mediated preeclampsia, that there would be a role for that kind of testing, and that the hope is to possibly prevent the development of disease, especially in patients with a history of preeclampsia, and that they are at higher risk for preeclampsia in a subsequent pregnancy. Because right now there are no drugs for the prevention of preeclampsia, which in many cases are thought to start in the first trimester with impaired placental invasion.
Okay, so correct me if I'm wrong, Wendy, but what you've said is that hopefully it's possible that the way that this could look in the future is that at the 10-week non-invasive prenatal testing test time, when the mother gets the blood drawn to find out the sex of her baby, and also whether the baby is chromosomally normal, whether it's a euploid embryo, at that time there would additional blood may be drawn to see fetal genotype. And at that point, if fetus has one or two variants, mother would be put proactively on this medication that you will have discovered. Fingers crossed.
I mean, that would be the ultimate ideal situation.
Yeah.
Although, like I said, treating somebody who has a known prior history of preeclampsia would definitely be something that might be considered. Whether or not someone is. Whether we wait for someone to show some signs of preeclampsia, if there are early preeclampsia tests before you treat with a drug, versus treating before someone even evinces any signs of preeclampsia, that would be kind of dependent maybe on the risk profile of the drug and other issues that the patient has.
Okay, so what you're saying, maternal history is also very important. Technically, yes. We don't have to wait for the blood test that shows fetal genotype. Let's look at maternal history. If she had a previous preeclamptic pregnancy, is her mother's preeclamptic pregnancy enough to make her at risk of having preeclampsia?
Well, there are cases of familial preeclampsia, but I think that we just don't have a lot of knowledge about the genes that lead to preeclampsia. And then one other thing I wanted to mention is that, because the APOL1 variants are in a very specific subset of the population, like I said, patients of recent African ancestry. So I don't think, I think you'd want to be very specific on who you would treat with drugs that are targeted toward APOL1 variants, and not necessarily like the universal treatment.
Yeah, absolutely. Now, Wendy, we know that Black women in the U.S., African American women, face a 60% higher preeclampsia risk than white women in the U.S. Do you believe that this statistic is so high partially as a result of these APOL1 variants?
I mean, we because we know that the APOL1 variants are playing a role in preeclampsia in this population. I think that there's definitely a possibility that the APOL1 variants are contributing to the increased prevalence of preeclampsia in Black pregnant women, although we don't know exactly right now the specific contribution of the APOL1 variants as opposed to other factors.
Yeah, yeah. So, but as you mentioned that the variants likely account for about 12% of preeclampsia cases in African American women in the US, that's the highest genetic risk for preeclampsia that we know about, period. There's no other higher genetic risk. So this is the leading one. So it's clearly very important.
Yes. Yes, that's really one of the few genetic risk factors that we know of. Period. Just not just in this ethnic group.
Okay, at all.
Yeah.
For any, okay. So it seems then to be very important, very significant. Why has this not been studied more?
You mean genetics and preeclampsia?
Yes, yes.
Yeah,
the genetic factors of preeclampsia.
It's a great question, Alessia, and it's because that it's very difficult to study genetic risk factors in preeclampsia because typically you need to study two genotypes, maternal and fetal, and so that's challenging. And then you can also only study the patients who have either when they're pregnant or that of that develop preeclampsia in their pregnancy. So it makes it more difficult, to study this genetic factors. There have been genome-wide studies, looking at trying to pinpoint genes that play a role in preeclampsia. Most of these have been done in, Northern European countries where they have the large registries. So that's something that, hasn't been studied in all populations.
Yeah. Couples undergoing IUI or IUF, IVF often take genetic tests before attempting to conceive, or even couples not undergoing IUI or IVF, yes. Just couples wanting to naturally conceive, want to see if they've got any gene disorders in either partner. Is the APOL1 variant included in any commercial testing platforms that could then say, hey, my partner's got a variant, I've got a variant, that means my baby can have two variants?
Yeah, I mean, they're not, the APOL1, as far as I'm aware, isn't included in any of the preconceptional commercial testing platforms. I believe that there are some tests now for patients with kidney disease. Patients are now being tested on commercial platforms, but there hasn't been any preconceptional testing of patients looking to get pregnant. That, like I was saying before, that might change at some point.
Yeah. Okay, so we've now talked about what we know about APOL1 variants, what we know about their ability to influence preeclampsia risk and to get preeclampsia. Let's talk about the research that you're going to be doing, which is to hopefully find drug compounds that decrease stress in cells, preeclamptic cells, which means essentially lessen the impact of their preeclampsia and don't lead to full-blown preeclampsia and the very serious adverse outcomes that come with preeclampsia. So you're looking to discover drugs to combat preeclampsia in your lab, Wendy. So let's talk about that. So the way that you're going to be doing it is, and you've already done this, you have made 6 placental cell lines, right, Wendy? You've got 2 normal placental cell lines, 2 G1 cell lines, and 2 G2. And I visited your lab several weeks ago, and I saw this. And you basically have these geometrical petri dishes that have a lid, and you've got liquid in each of them, and you've got 6 of them, and they're quite small. And at the bottom, you have these invisible cells that you can see through a microscope. And they look kind of like specks of dust, and they're all kind of different shapes. And it's interesting, they stick to the very bottom of these petri dishes, right? And you keep them in like a microwave to make sure that they're nice and warm and cozy and that they continue to grow. And you feed them good nutrients, and you give them even antibiotics to make sure that certain bad bacteria are killed off, and only the ones that carry the DNA will come to the forefront, and all sorts of very cool stuff. So, but essentially what you'll be doing, tell me what you'll be doing with this assay. So you've got these 6 dishes, Wendy, and tell me how you're going to test these different cells to see how they react to these compounds that you're going to put on them in several months. I think over the summer and over the fall, you're going to be doing this work. So tell me what that's going to consist of.
Yeah. So yeah, it was great to have you in the lab the other day to look to see our setup. But yeah, so essentially we have three cell lines. We had them in duplicate, so you saw 6 flasks. But we have 3 cell lines that we made. We made them from. We used a placental choriocarcinoma cell line as the base, called BWO cells, and we did something that's already been done in kidney cells when kidney researchers were studying APOL1 in the kidney cells, which is we introduced in those cells that don't express much APOL1 on their own, we introduced into them DNA that expresses APOL1. So one of the cell lines expresses the normal or wild type APOL1, the G0, we call that G0. And then the other 2 cell lines express one expresses the G1 variant.
So you basically injected in them this normal and abnormal DNA.
Yeah, we use something called lentiviral infection to make sure that the cells always express that DNA. Okay. So one of the cell lines, we call it the G1 line, expresses the G1 variant, the G1 APOL1 variant, and then the other cell line expresses the G2 variant of APOL1. So those cells, and then the way that we have it, the cells expressing is that it's under control of a tetracycline, what we call a tetracycline inducible promoter, or a TET-on promoter, where when you treat the cells with tetracycline or another antibiotic like tetracycline, like doxycycline, that turns on the gene. So we can turn on the gene when we want to do an experiment, because having the gene on all the time might kill the cells if you had it on all the time.
Okay, so to use an analogy or a metaphor, Wendy, you've got these cells. They start normal, even though they are tumor cells. They're placental tumor cells, but they start normal. You essentially, for lack of a better term, inject them with a light bulb of DNA, and then you put an antibiotic on top of them to turn them on. As a light switch, you turn on the light switch, the light bulb turns on inside, and it begins expressing and behaving like a G1 or a G2. So now that's the foundation, but the exciting part comes when you sprinkle these compounds on top of these cells that are already expressing the variants. So they're behaving like variants, like the APOL1 variant. You sprinkle the compounds on, and you watch to see which ones light up bright green, right?
Yeah. Well, so that's, first of all, I love your light bulb analogy. So that's a great way to express it and to try to help people understand, what's actually going on.
It's complicated stuff. And I remember the last time we talked about this, I said, Wendy, why has this not been studied so much? You said, it's extremely difficult. You said for many reasons, it's complicated and difficult. And so I've got to do the light bulb to understand myself.
I mean, trying to explain it is always something that we want to make sure people understand.
Yeah.
And so basically so kind of like you were saying earlier in the discussion, it's kind of a 2-parter. First of all, the reason we select the placental cell line as the base, and immortalized placental cell line as the base, is because once the variant is turned on, we want to study placental cell behavior, or how the placental cell functions are affected by turning on the variant. So these are placental cell functions are, there are various ones, such as placental cell fusion or HCG, placental hormone production, things like that. The other thing that we want to do is identify, as part of the research that the grant is funding, is to identify ways to detect, cell dysfunction in the cell when the variant is turned on, and then to capitalize on that to develop an assay in order to look for drugs that can decrease or improve the negative effects of the APOL1, the turning on of the APOL1 gene.
Yeah.
So that is, so one of the proteins that we proposed looking at was a cell stress protein called GRP78, but there are also other cell stress proteins that could be increased, their levels could be increased by turning on the APOL1 variant. And so based on one of those proteins, we want to develop the assay, which you were saying, the glowing, the fluorescent, based on that, we want to develop a fluorescence assay that can show us which drugs can decrease that, those adverse effects or increases in cell stress that are initiated by turning on the variant or the light bulb, like you were saying.
Can you see, I think I asked you this question before, but I'm not sure.
Mm-hmm.
Can you see the cells become stressed? Is this a visual thing you can see under the microscope, or it's not really? It's just that when you add that tetracycline,
Mm-hmm.
when you turn on those variants, they just become stressed and you cannot see that happen.
So that's a really good question, because sometimes people will look at things like cell shape and cell appearance after cells are stressed to see if they change in shape, or if they have maybe decreased cell fusion, which is cells that kind of come together as would happen in normal placental development.
Yeah. Even years ago, we would judge embryos by their morphology, right? So this is a great example.
Yeah,
just
well,
the way
we still do that.
We still do, yeah. No,
we still do that. Yeah, but the thing is, things like increased expression of a stress protein. Usually, you need to measure the protein levels either by doing like something like a Western blot, or
to see if it's high.
Like I said, or like the fluorescent assay, which can show that there's increased levels of a stress protein by looking at a special fluorescent reaction in the assay.
Okay, so the assay is going to show the green when there's stress, and then when you add the compound, it will show less green.
Yeah, well, the thing is, it's that particular, the color, it's not exactly green, but not like green fluorescent protein or anything. It's actually kind of probably more of a yellow color, but
okay.
But yeah, so basically, for example, if we turned on the APIL1 variant, or turned on the light bulb, as you said, if there's a drug that can diminish the shining of the light bulb, or turn it off,
I see.
Because in this case, turning on the light bulb is a bad thing. If there's something that can decrease the light bulb or the light from the light bulb, then this assay will show that.
I see. Okay, so you turn on the switch, you activate the DNA. There's light everywhere. There's stress everywhere. The cells are growing preeclamptic. We put the compounds on the cells, and they're going preeclamptic because of this one protein, and probably others as well. And then we put these compounds that target this protein and see which ones are actually effective at targeting it by watching which lights are getting dimmer.
Yes.
Okay,
that's a good analogy.
Okay.
That's a nice, simple way to think about it. And the thing is that, because we don't really understand, that no one really understands the root cause of why preeclampsia happens. So in diseases where you don't know the root cause, drug discovery and drug screening is one way to try to find treatments, because we can't necessarily explain like exactly what is happening in the placental cells to cause the preeclampsia. But if you can interfere with the detrimental process that leads to preeclampsia in a model such as this, then you have a hope of finding drugs, even if you don't understand the mechanism underlying the disease. So that's how drug discovery is so helpful in diseases where we don't know the cause.
Yeah. Well, based on that question, Wendy, essentially I'm going to ask you that same question that you already answered, because now I'm very curious. And this seems very obvious. Decreasing stress will likely stop the preeclampsia from developing. But how do we know that? Maybe there's other factors involved. Maybe this stress protein, hey, you damp it down a little bit, okay, but maybe preeclampsia still develops. And just being devil's advocate here, how do we know that stress is the key pathway failure here?
Absolutely, Alessia. That's a great point, and that's why it's so important that the drug screen that we'll do on the cells is really just the initial step, like the baby step forward.
Okay.
Once we find candidates, then we need to carry those drug candidates into an animal model. And like I told you, previously, we have collaborators that have both preeclampsia mouse models that are unrelated to APIL1, and then we also have APIL1 variant mouse models that we can use to test the candidate hits. We call them hits that you get from the drug screen, to see if they will interfere with preeclampsia in an animal model. So that needs to be done before there's any move toward translating that to treating humans.
Yeah. And the other important thing is you also want to make sure that there's no adverse effects, unintended consequences from these drugs. Maybe they do, in fact, tamper down the stress, they stop the preeclampsia from developing, but they have other unintended effects. And so you also, the mouse model is important for that reason too, right?
Absolutely.
Side effects,
Absolutely.
essentially side
Right.
effects.
That is absolutely an excellent point. And although the one thing about the APL1 is that in terms of things that can to suppress APL1, the concern is always something like, would you interfere with normal pregnancy or normal bodily functions if you just canceled out APL1? And we know there have been patients identified with complete humans with complete knockout of APL1, and they survived. They had no issues with survival. So we know that at least in the area of APL1, if that was completely inactivated, that there would be the patients would still be able to function relatively normally. So that's one.
Okay, well
that's appeal of this area. At least we know that it's not required for survival. So
fantastic. Well, that's great news. So that begs the question: why on earth do we have it as a gene?
Well, we do know that one of the roles that it plays is it's a carrier of HDL, so it's a like many apolipoproteins we have.
And that's a good fat, right? That's the good cholesterol.
Yes. So there are other functions that it serves, but there may be redundant functions that other apolipoproteins can also assist with.
Okay. You talked a couple minutes ago about how we don't exactly know the pathway of preeclampsia. That's why this work is so important. We don't know exactly what's causing it, but we do know the placenta is absolutely vital. Tell me what we know about how preeclampsia affects the placenta, and then how that has then downstream effects on the woman, the baby, different organs. I think you mentioned that the primary way in which it interrupts is it essentially makes the placenta less sticky to the endometrium. So the placenta is not as attached to the uterine wall as it should be, and as it is in normal pregnancies. And this lack of Velcro-ness prevents nutrients coming from the mother to the placenta to the baby. And then the placenta does all sorts of crazy things to make up for it. And this leads to essentially illness.
Yeah, I love your analogies, Alessia. The Velcro analogy is great.
You need a great teacher to be a good student, Wendy. So it's all on you. It's all on you.
Exactly.
You're the teacher.
Yeah. Well, the thing is with preeclampsia, we know that that there's something causes impaired invasion. That in early pregnancy, the placenta has to invade into the mother's tissue or decidua, so that in order to access. Invasion. It's actually a good invasion because the baby needs to access maternal blood supply, or the oxygen. Absolutely. So in preeclamptic pregnancies, the invasion is not well developed. Like, it's not complete. The invasion is very shallow. So
do you think the mother's immune system has a role there?
It's a possibility. It's a possibility because definitely. But we do know that people are actively researching these areas. But we do know that the lack of the poor invasion of the placenta and kind of the resulting like low oxygen can lead to the fetal growth restriction, smaller placentas, increased risk of something called abruption, or separating of the placenta that's not well anchored in the maternal tissue, is early separation that can lead to adverse outcomes with the pregnancy. So there's, and then we also understand that with preeclampsia, that there's probably some maternal response to the placenta trying to access maternal nutrients, and later on the factors released by the placenta can lead to maternal high blood pressure, loss of protein in the urine, swelling, things like that. So there's
so it's quite sad actually, right? Because the placenta is trying to save this pregnancy, and in excreting in its efforts, it's actually hurting the pregnancy. with these factors that it excretes. Well, it's certainly a fascinating topic, and the placenta itself is very interesting. So we don't know what who is the key player, Wendy, in that invasion, because maybe we can strengthen the army in that
Mm-hmm.
invasion to to
Mm-hmm.
to better invade. I don't know how we
Mm-hmm.
do that, but
Yeah.
maybe we think about the front lines.
Right. Well, it's definitely so we think it's something to do with the fetus, because the placenta comes from the fetus, and
Yeah.
so that is what is responsible
Okay.
for invading, and that's what makes the APL1 variant story so compelling, is that we know that it depends on the fetus APL1 variant.
Yeah.
So
Yeah.
that totally
Yeah.
is consistent with this story that that preeclampsia is a
Yeah.
disease of the placenta and poor placental invasion. So
Yeah.
that's all consistent.
Yeah. So maybe the theory is that stress makes the invaders weak or something, and they cannot invade properly into the endometrium.
Well, in terms of what we know of the APL1 variant role in kidney cells, is that
Yes.
they've shown that APL1 variant expression in kidney cells, the abnormal variant expression increases cell stress and that's been shown in things like increased levels of GRP78, which is a cell stress protein, as we talked about earlier. We're basically trying to kind of translate those kidney studies into the placenta, because that would help us, number one, like we were talking about, understand the mechanisms by which preeclampsia affects, or at least the APL1 variants affect the placental cells, and secondly, may help us to screen for drugs that could be beneficial in preeclampsia.
All right. So now, Wendy, this is the first time that this type of preeclampsia has been studied in a placental cell line, right?
Well, I mean, preeclampsia in general has been studied in various ways, and people have tried to look at,
But
different, cell
APOL one.
models. But yeah, this is probably as far as I know, this is the first cell model which has employed APL1 as in this fashion,
Yeah,
to
yeah,
use as a drug
yeah.
screen, and also to look at mechanism.
And tell me how many drugs you're screening here. What is it? Something like 1,600 drugs or something
Yeah,
like
well,
that?
we have in the initial screen, we'll probably be looking at our we have a collaboration with the Boston University Center for Molecular Discovery, and they have um, a small library of about 4,000 representative compounds, and then there's also we also have an FDA approved set of compounds that's commercially available. That's probably about 1,600 compounds. So that'll be kind of the beginning. And then these are, just want to point out that these are, even if it's not like millions of compounds, like some of the pharmaceutical drug screens, it's a very highly curated set of compounds with representative compounds from different, chemical classes. So it's going to be kind of like, like a kind of quality over quantity type screen.
And have all these drugs been shown to have some relationship with GRP78?
Well, no, they have different functions and not necessarily related to GRP78, but that's kind of with a drug screen, you're casting a wide net to look at different drugs that could possibly have action, to
You
kind of
want to
try to
you want
correct
to
the
you want to put some unlikely players in there, right? To see if
exactly, yeah,
there's some surprising effects that happen.
absolutely.
Okay. Now tell me about the second or first, depending on how you look at it, part of your
Mm-hmm.
research. So one is the drug screen, and the other one
Mm-hmm.
is actually watching what effect APOL1 variant expression is having on these cells. So what effect
Mm-hmm.
is the variant having on the development of normal placental cells
Mm-hmm.
to abnormal? What are you
Mm-hmm.
expecting to see here, Wendy? And
Mm-hmm.
what findings could come from this?
Mm-hmm.
What
Yeah,
aha
that's a
moment can
great
come from
question.
this?
Yeah, it's a great question, Alessia. And like you were saying before, turning on the light bulb and then to see what happens when the light bulb is turned on, when we're able to activate that selectively activate the variants. So things we can look at with the B well cell line in particular includes things like we were talking about cell fusion, which is required as part of placental development. We call that syncytialization. So we can look at differences in that, as well as differences in HCG hormone, pregnancy hormone production, which is also critically important and something that we observe in the early pregnancy. So those are just some of the tests that we can look at to assess placental cell behavior, and seeing how turning on the light bulb will affect those normal behaviors.
Yeah, absolutely. And now, but this is all specific to the APOL1 variant, right, Wendy? So are we going to be able to, are other scientists and are you going to be able to take learnings from this specific phenotype, from this APOL1 variant phenotype, and say these are general statements we can make about preeclampsia? Can we make some more broader, can we learn some broader lessons about preeclampsia from this very specific niched phenotype work?
Yeah, well, that's the hope, is that if we can show that perhaps cell fusion is interfered with, or there's some issue with the HCG is impaired, production is impaired when we turn on the light bulb, that might inform us other research looking at preeclampsia and saying, hey, this is something that is affecting placental development at the very earliest stages, and how can we study that further, so that could possibly shed light on the earliest origins of preeclampsia, and that's what we're hoping for.
Yeah, absolutely. Okay, Wendy, as we conclude, I want to get into some timelines here. When we can expect to see something come out of this research? How long do you think? When do you think you can have a short list of compounds that are promising? And then what would the next steps after that be?
Yeah, well, that's again a great question. And so in terms of assay development, we're going to be, like I told you, this over the summer and possibly early fall, we'll be trying to identify the best candidate to use in the assay development, whether it be GRP78 or other cell protein, stress protein. So we'll be
Can you
using
use
that.
multiple in the same assay, or no? Can you use multiple
well,
proteins?
typically just from the cost of the assay, usually want to try to hone in on the best one, and then you can, we can identify that by doing, Western blots or other tests to see, to optimize before we go into the assay development.
Okay.
And then we'll be working with Revity, which is a company that we've worked with before, which specializes in making assays like the homogeneous. They'll be helping us with an assay called the homogeneous time resolved fluorescence assay, HTRF assay. They set their timeline at approximately 2 to 3 months for development of the assay, and then we would want to do kind of secondary verification of the assay in the lab prior to starting the compound screening. And the compound screening itself is, it's designed to be high throughput, can be done in 96 well plates or 364 well format. So the idea is to try to set up the assay, cell-based assay that can be done rapidly with many compounds at once.
Fantastic. So when do you think we could have a short list of drugs, Wendy? You said 3 months for the development of the assay. Let's say the assay is developed in the next 6 months. Maybe.
Yeah. Well, we would hope that at the end of the granting period, which is probably 2 years, we're hoping that we could have a list of candidate compounds.
Yeah.
That would be ideal. Yeah.
Very cool. Very cool. And so, Wendy, do you think that your research will lead to a new medication for women that are at risk of preeclampsia?
Yeah, I mean, as scientists, we always hope that our research will yield to a breakthrough or some, provide more scientific information to help patients, who are suffering from illnesses and disease. So that's our our hope that what we do, that our work will lead to improved treatments, or at least kind of open the discussion, because right now we don't have any effective treatments for either preeclampsia treatment or prevention, because things like magnesium and other treatments are basically to treat the sequela or the findings from preeclampsia, like to decrease the risk of seizures and things like that, and not necessarily to treat the root cause of the disease, because we don't know what the root cause is necessarily. So
Yeah, including aspirin.
So the thought of the aspirin is that there's so possibly some ways to help with things like maybe improve placental invasion, things like that. So but the problem is that because we're a little bit in the dark as to the absolute like what is the cause, the actual cause of the preeclampsia. We're kind of limited to treating. Yeah, we're kind of limited to treating the findings of the preeclampsia.
Yeah. Do you feel a little bit like the more that you find out, the more questions you have, or do you really feel as if though you guys are going in the right direction here with the invasion part?
Yeah, I mean that's definitely how research is. Like the more you find, the more questions come up, and that's one of the great things about doing research as a physician scientist is that it's kind of a continuous process of discovery, and you're always finding new things.
Well, Wendy, I hope that you find something new and something wonderful here with these compounds to help all the women that are suffering from APOL1 related preeclampsia, not only in this country, but in other countries, and also some general findings for women that are suffering from other phenotypes of preeclampsia. And it's been an absolute pleasure talking to you today. This is really exciting work. Come back the minute that you have your short list of candidates, and we want to hear all about it, Wendy. So thank you for joining me today.
Oh, thanks so much, Alessia. And as I've said before, I think it's so important to have what you're doing with the modcast and introducing science to the general population and to lay people, because I think all too often scientists don't do enough, don't communicate their findings or their research to the general public, and that's, I think it's so important for us to let the public know what we're doing and why we're spending the dollars that go into research, and the importance of research funding.
Well, it's absolutely my pleasure and the pleasure of my team and our chief scientific advisor, Dr. Emery Selly, who pushed for this podcast from the beginning. It was his idea, so all credit goes to him. And this show would be nothing without you, Wendy, and all the scientists that we have on it. And our greatest pleasure and joy is just to shine the spotlight on you and the incredible work that you're doing, truly. So all the best, and I hope to see you both in your lab and on my screen very soon.
Thanks for having me, Alessia.
Thanks, Wendy.